Before polymersomes came into the picture, drug delivery was a bit like trying to send a fragile gift through a violent storm. Scientists had these incredibly delicate drug molecules that needed to reach a very specific destination inside the body. But the bloodstream? It's chaos. Enzymes waiting to chew things up, sudden pH shifts, immune cells acting like patrol guards. It's a hostile environment. For years, we relied on liposomes, fat-based vesicles, to carry drugs. They helped, but they weren't perfect. They leaked easily, broke down fast, and simply couldn't withstand harsh biological conditions. So scientists asked a simple but powerful question. What if we built something tougher, something more customizable, something smarter? From that question, Polymersomes were born. Engineered mini-capsules designed to deliver drugs, proteins, and even genetic material with precision and strength. So, what are polymersomes? Think of them as tiny synthetic bubbles, but engineered with purpose. They're made from amphiphilic block copolymers, which simply means polymers that have one block that loves water and another that avoids it. When these polymers touch water, they begin to organize themselves automatically. The water-loving parts face outward, the hydrophobic parts hide inward, and together they form a hollow spherical vesicle, a little container with a sturdy membrane. They look a lot like a cell. But unlike natural vesicles, polymersomes are customizable, stronger, and far more stable. They aren't just structures, they're intelligent carriers built by design. To understand why polymersomes are gaining so much attention, we need to compare them with liposomes. Liposomes have thin membranes, almost like soap bubbles. They're biocompatible, yes, but incredibly fragile. Polymersomes, on the other hand, have much thicker membranes, anywhere from 5 to 50 nanometers. That thickness gives them higher mechanical strength, better stability in the bloodstream, longer circulation times, and significantly reduced leakage. If liposomes are soap bubbles, polymersomes are like high-end sports helmets, engineered to survive impact and keep their cargo safe. That robustness makes them ideal for delivering sensitive or potent therapies. Polymersomes get their properties from the block copolymers used to build them. Common examples include PEG-B-PLA, PEG-B-PCL, PEG-B-PBD, and PMPC-B-PDPA. Each has a hydrophilic block, often PEG, and a hydrophobic block that determines how the vesicle behaves. The polymer you choose affects almost everything. Degradation rate. Some break down slowly, others quickly. Flexibility. Softer polymers make more fluid membranes. Responsiveness. Some react to pH or temperature. Drug compatibility. Hydrophilic versus hydrophobic loading depends on the material. Choosing the polymer is like choosing the DNA of your vesicle. It defines how it will behave in real applications. One of the most incredible things about polymersomes is that they practically build themselves. When you mix amphiphilic polymers in water, they naturally organize into vesicles. Hydrophobic blocks cluster inward, hydrophilic blocks stretch outward, and the system settles into the most energy-efficient shape, a bilayered sphere. Scientists can control this self-assembly through several methods. Changing polymer ratios, using solvent evaporation, hydrating thin polymer films, or using microfluidics for precise size control. Microfluidics, in particular, has revolutionized this field, letting researchers produce polymersomes with uniform sizes ideal for drug delivery. Self-assembly isn't just chemistry, it's nature choosing the most comfortable configuration. One of the biggest advantages of polymersomes is their ability to carry a wide range of therapeutic molecules. They're often described as the Swiss Army knives of drug delivery. They can load hydrophilic drugs inside the aqueous core, hydrophobic drugs within the membrane, proteins that need structural protection, DNA or RNA for gene delivery, imaging agents for diagnostics, and even enzymes for catalytic functions. This dual loading capability gives polymersomes a versatility that few other delivery systems can match. Now, here's where polymersomes truly become smart. They can be engineered to respond to specific triggers inside the body. Imagine a vesicle that stays completely silent and protected while circulating through the bloodstream. But the moment it reaches a tumour's acidic environment, it opens and releases the drug exactly where needed. That level of precision is possible with stimuli-responsive polymersomes. pH-sensitive, for tumours or inflamed tissues. Temperature-responsive, useful for hypothermia treatments. Redox-responsive, activated inside cells. Enzyme degradable, breaking down only in specific organs. These are not just carriers, they're programmable delivery systems. Polymersomes are rapidly moving from academic curiosity to real-world applications. You'll find them being studied in cancer therapy, gene delivery, immunotherapy, vaccine design, biosensing, diagnostic imaging, artificial cell systems. One of the most futuristic uses is the development of artificial organelles, synthetic compartments inside living cells that perform specialized tasks. This could revolutionize synthetic biology and redefine what cells are capable of. Of course, polymersomes aren't perfect. There are important challenges researchers must account for. Polymer purity. Even small impurities can disrupt vesicle formation. Drug loading compatibility. The drug's chemistry must match the polymer. Stability versus degradability. Too stable and the drug won't release. Too degradable and it breaks down early. Scale up. Microfluidics is promising, but industrial production is still tricky. Regulatory pathways. Because these are synthetic nanomaterials, they require rigorous safety evaluation. These challenges represent active research areas and opportunities for innovation. Polymersomes aren't just tiny containers, they're platforms that could transform the future of medicine. As polymer chemistry advances, we're seeing vesicles that are smarter, more precise, and capable of tasks we once believed were impossible. They're pushing boundaries in precision cancer therapy, gene editing and delivery, next-generation vaccines, advanced immunotherapies. For anyone entering polymer science, drug delivery, or nanotechnology, polymersomes are one of the most exciting frontiers to explore. The future is bright, and polymersomes are going to play a major role in shaping it.